Okay, everyone, welcome back. This is our next lecture on microbial metabolism. Just a fair warning, I expect this one to be rather long, and the next week's one on genetics will also be, I anticipate, quite long. But we'll see. We'll see how it goes. So, first things first. All living things, whether they're prokaryotes or eukaryotes, are going to have to do a lot of the same kinds of actions in order to stay healthy, to actually grow, and to get to the point where they're ready to reproduce, and also to do that process. And this includes maintaining homeostasis, which that is a whole can of worms unto itself. Many different tasks involved in maintaining homeostasis. But in the process of this and these other processes, they're going to be making lots of new molecules, whether they're building proteins and enzymes or individual building blocks for those things, or maybe they're making larger structures that are not protein based, like phospholipids for the cell wall, for example. Or maybe they're even making defense compounds or toxins to help them survive in their environment or do certain aspects of their ecology. At the same time, these organisms will also be breaking down molecules. They'll be taking things into their body and digesting them, breaking them up into their raw materials, and then recycling things that they've already made at times. When they're done with something, that stuff doesn't just get thrown away. It often gets recycled in some way to turn it into something else that can be useful, which is much more helpful to this, what is sort of a closed system at times, a cell, that you don't have to go and acquire a bunch of new resources. You can use the stuff that you already have. In addition, these cells are going to be making lots of ATP and utilizing a lot of other energy sources. Depending on what we're talking about, we might be looking at inorganic sources like hydrogen sulfide for some of these bacteria that grow on like deep ocean vents, for example. Other species might be using something very different like nitrates and nitrites and breaking those apart into atmospheric nitrogen. Other species are going to be using organic sources of energy like glucose. And they will often be getting that stuff. They'll either be making it themselves from some other source, or they will be capturing it from their environment or from other organisms, like a predator would. And of course, there's also a lot of internal organization that is going on in this process too. The cell is not just a big, jumbled, disorganized mess. There is a method to that madness, and the cell will organize its internal components accordingly. And many more things. There's lots of different tasks that fall under this umbrella. Now, all of these jobs ultimately require metabolic activity in some form or another. When we talk about metabolism, what we're really talking about here is the chemical processes that an organism will do in order to maintain life, homeostasis, to do their reproductive pathway, different things like that. Okay, and metabolism is super broad, but it can be categorized into two main groups. You have catabolism, which is what you might call destructive metabolism. So you take a larger molecule and you break it apart into its smaller components. And then you also have anabolism, which is constructive metabolism. Now we're taking those smaller components and we're building larger, more complex molecules from them. And every cell is going to be performing both of these processes across thousands and thousands of reactions, of different types of reactions. Of course, they're doing this on such an incredible scale. We're talking about millions and millions of reactions that may happen in a very short period of time. But there are thousands of different kinds of reactions that a cell may be performing along what we call regulatory pathways. So this is all part of this large umbrella concept of a metabolic pathway, which essentially means a chain of chemical reactions to go from one state of one or more molecules to a final state where we have produced a molecule of that the cell has sort of intended to make, whatever that final product looks like. If we're talking about like a human cell, a final product for this sort of metabolic pathway may be something like insulin. And then the process of the metabolic pathway for making insulin may be, that'll include the transcription of DNA into RNA, the translation of RNA into that protein, and then also the rearranging and forming the final structure of that protein. So, and there's several steps involved there. But there's lots of other things that may be subject to a metabolic pathway, and we'll sort of explore some of those things later. I want to stress it's not just something that we build proteins with. This is for basically everything that a cell will make for itself. So ultimately, when we talk about a metabolic pathway, we're talking about a multi-step process. Vast majority of metabolic pathways are many steps, where you start with this starting compound, whatever it may be, and you have one reaction that makes it into an intermediate state. And then a series of additional reactions that may change that intermediate state to something else, until eventually you get to the final reaction that turns it into that end product. Okay? And some important terms that I want to lay down for you as we explore these subjects, as we talk about enzyme activity and metabolism, is first we have substrates. Okay? A substrate is a reactant, a chemical or molecule that is going to be reacting in a metabolic reaction. Those intermediates are those middle steps in a reaction. So they're those intermediate positions in the metabolic pathway. The product is the is the final result or results of a metabolic pathway. Whatever the sort of intended goal is for that cell. That may mean the final molecule that is going to go off and become some sort of, maybe it's a toxin. Let's call it a toxin. It goes off and does some sort of bioactive role in a disease-causing bacterium. But there may also be byproducts and waste materials that come from these additional reactions. Things that were sort of not the intended goal, but they are made in the process of making that intended product. And by the way, those things can be, they'll either be removed, sent out of the cell, or they'll be recycled and repurposed into other things. And then we have enzymes. And enzymes are really important. These are the active moderators of these metabolic pathways. We call them a catalyst. Okay? A catalyst is something that facilitates a reaction, a chemical reaction, without itself being used up. Okay? Now, because these metabolic pathways are predominantly many steps, there's very few of them that are just like a single step. Most of them are many steps. We are going to have multiple enzymes that facilitate each of those independent steps. So you really are going to have this system of enzymes that operate these large metabolic pathways, each one of them controlling one important step in that chain. And these enzymes, they vary a lot in their jobs and what substrates they react with. So you're going to have a different enzyme for step 1 of a metabolic pathway, step 2 of a metabolic pathway, step 3, and so on, all the way down, because they are so specific in the jobs that they do. And so that's really because these are lock and key mechanisms, effectively. We can think of them that way, where imagine the enzyme is the lock and the substrate is the key. That there really is only one or maybe a few shapes of a substrate that will fit that one enzyme. that one specific type of enzyme. So only certain substrates are actually going to be interacting with certain enzymes. Enzyme 1 will only take the starting compounds for this metabolic pathway, react them, and produce intermediate A. Enzyme 2 will only take the intermediate compounds produced from this first reaction. They are incredibly specific. And it's kind of important to talk about why we have these enzymes, and we'll talk about this again in a little bit, but enzymes really just make the job easier. So these different metabolic reactions, in theory, they can happen in a cell or in a body without any assistance from an enzyme. But the amount of energy that they often require in order to make that happen is much greater than if they used an enzyme to do that. Consider this. Consider an enzyme kind of like a person that takes one thing and brings it closer to another thing. It pushes these two molecules together or bends a molecule in just the right way to create the conditions that are more conducive to reaction, to binding together two things, or to break something apart, or to change something in some way, maybe to twist it and change the orientation of parts of a molecule. There are many different things that an enzyme might do. And essentially, they really are just making that job easier. Their job is really just to put things in the right place for a reaction to happen. They set things up very nicely so that you don't need things to happen randomly, and you also don't need the considerable amount of energy that is needed at times to make these reactions happen on their own. When we talk about enzymes, it's pretty easy to identify in a sentence or in text, because you're almost always going to see the suffix "ase" afterwards, where the suffix "ase" is going to denote that enzyme. Okay, so there's lots of different examples of this. The names of these enzymes are also going to be really important to inform their function. So we can even, most of the time, figure out what an enzyme does. In the most basic of terms, at least, what it does by its name. There are a few enzymes that have less easy names that way. Many more that have very, very intuitive names. So, for example, lipases. Okay, lipases. Lip is in lipid, right? And so a lipase is something that will break down lipids. Proteinases will break down proteins. Nucleases will break down amino acids. You'll have other things like synthases that will synthesize something. Polymerases that will polymerize something. And by that I mean literally making a chain of the same thing over and over again. So a starch, for example, is a chain of carbohydrates. So there are some enzymes that may be polymerases for sugars, where they will bring sugars together and chain them together, right? We are actually going to see polymerases pop up again in the next chapter because they're responsible for polymerizing DNA. We also can find things that are more specific to the type of reaction that they do, like oxidases, which will oxidize something, dehydrogenases that will dehydrogenate something, as in take hydrogens away from it, pull them off it. Kinases are they're enzymes that will move a phosphate from an ATP molecule onto something else. That's a kinase molecule. A phosphatase molecule is going to attach a phosphate, not necessarily from ATP, but from something else. I don't want to go down a huge hole on this, because there are so many different names, but I wanted to explain that to pay attention to the names, because sometimes you can infer what the enzyme does, not just that it is an enzyme, but also its function from its name. So as I mentioned before, enzymes, when they bind, when they like interact with specific substrates, they have a lock and a key type mechanism where they're extremely specific. A specific enzyme is only going to associate and bind with specific substrates. This is very much like a padlock needing a specific key in order to open it, right? The substrate has to have a specific shape in order to match the unique shape of the enzyme. And so there's really only a few things of that shape that will fit inside that enzyme. I want to stress that there isn't necessarily one key for every one lock. There may not be the kind of situation that you might expect where there's literally only one possible thing, one possible type of substrate that will react with this and bind with this specific enzyme. An enzyme is looking for a specific shape, and if there are many things that have at least partially the same shape, they may be able to bind here. And in fact, that is what we see kind of broadly, where some enzymes, can bind with things that are not the target substrate necessarily. That will come back up a little bit later, but I want you to remember that. Once the substrate and the enzyme bind, they form what's called a complex. Okay? A complex is just both of these things bound together. So now, once they're bound together, they may be able to react and create whatever the intended product of that reaction is. But some enzymes need a little bit of assistance. Some do this without any assistance whatsoever. So they don't need to be modified at all in order to function. They just kind of do their job. But a lot of them do need a sort of assistance or modification in some way. So for those that do, they may need to bind to what's called a cofactor, in order to reach the shape that will bind with the specific substrate. So they need to get the right shape. I like this image here because it's kind of easy to visualize that, where you have a cofactor that will come in and bind, and together the cofactor and the enzyme will create the shape needed to react with a specific substrate. So these cofactors can be a lot of different things. They might be an inorganic ion like iron or magnesium or something like that. They come in, they bind, and that changes the shape. Or it might even be something larger like a coenzyme, a whole different enzyme whose job it is to come in and react with this other enzyme and work in tandem. Or it might be an organic vitamin, like a larger molecule that comes in and fits into this slot. Okay, so an apoenzyme. This is an important term. This is an enzyme that by default is inactive. It is inactive until a cofactor comes in and binds to it. So on its own, doesn't do anything. And it needs to have that cofactor come in and attach to it in order to achieve its function. In doing so, it changes the shape that allows the enzyme to now fit the substrate in its binding site. Okay. Without it, the shape is distorted. It doesn't, these molecules won't come in and bind. They won't interact whatsoever. It needs that additional cofactor in order to do that. Only together will they bind to the substrate. Now, cofactors typically have to be acquired from our diet, from the organism's diet, whoever is making it, whoever's making that enzyme. So this is very much the case for the iron and magnesium and zinc and stuff like that, and also those vitamins, those specific vitamins. But some coenzymes can be synthesized by certain organisms. Sometimes we have to get them from other sources, but some organisms can make them. Okay, the coenzymes that are either a protein itself or a vitamin. So this is something that some bacteria can do actually. So like E. coli, for example, is capable of doing this for some of these these vitamins specifically. So it's a quality that varies a lot from species to species. Some will be able to do this regularly. Some will that will not be a factor of their species, and they have to be able to get them from their environment. And here we have a sort of similar pattern of nutrient cycling that's going on here. If you think back to 2 chapters ago, right? That some of these organisms are able to synthesize their own cofactors, and some of them aren't. The ones that do synthesize their own cofactors are making these cofactors that can then be acquired by other species when they die or are eaten in some way, or they can be traded perhaps, different things like that. Okay? So here again we see the reorganization of nutrients and the way that might influence their sort of availability, right? Okay, now we also have enzymes that can be changed, not necessarily in the way of like starting off non-functional and becoming functional, but a functional enzyme that can be enhanced or inhibited by adding different things to it. That they have these additional binding sites on it. So what we're talking about here is an allosteric enzyme. An allosteric enzyme. So an allosteric enzyme is an enzyme that has an allosteric site on it. An allosteric site is a secondary binding site. It's not what we call the active site. The active site is where the substrate is going to come in and bind. Okay, but a secondary site that is not the active site, where what we call an allosteric modulator will come in and bind. And the modulator could be an inhibitor, which means that when it binds to the allosteric enzyme, it changes the shape in a way that prevents the substrate from binding. So it'll transform the active site into something that now doesn't fit the substrate. Okay? So it comes in and it can't bind and it kind of goes off on its own, right? No reaction happens. Or we might be looking at a promoter instead, which alters the shape of an enzyme to encourage binding, to make it easier for the substrate to come in and bind. Okay? Important to note, unlike the apoenzymes, these modulators are not required for an allosteric enzyme to function. Okay? They don't need it to function. It will do basic function on its own, but it could be enhanced or inhibited with the addition of these modulators. So let's talk now about temperature. We're going to start talking about some factors that influence enzyme activity. Okay, we've already kind of explored this in previous units where we've talked about how like in the last unit that proteins, enzymes can be denatured under a certain temperature. If you take the temperature up too far, the enzymes they stop working and they undergo denaturation. Okay, enzymes have an optimal temperature for function, and that optimal temperature, it can be one thing, and then you'll have a range of possible function across a wider range. Okay, wider range of temperatures. So the optimal temperature here for this one is say 47 degrees Celsius. Okay, but it can still function at temperatures all the way from around 55 or so degrees Celsius down to below 10 degrees Celsius. Now, at these temperatures, the function is incredibly limited. All the way down here, really not functioning that well. Over here, really not functioning that well. Past this threshold, this optimal rate, you start getting the temperature, interfering with the shape of the enzyme again, and you get denaturation. Okay, the denaturation is changing the shape of the enzyme, and it's kind of losing its ability to bind with the substrates and do its job. Past a certain point, all of these enzymes have been denatured, and there's no longer any function. Okay, all the way down here at the far cold end, you're not really getting denaturation, but molecules are moving much more slowly, and there's a much reduced rate of encounter between the enzyme and whatever it needs to react with. Those reactions need a certain temperature in order to function sometimes, and so they really decline in their sort of ability to do those reactions. Anyway, it's important to note, to get to know this chart, right? Get to know this chart, and also the relationship here between temperature and an enzyme activity, right? The ability for these enzymes to do their reactions. So, of course, because we see different species living in some pretty different habitats, you'll have psychrophiles that love to live in very cold environments. And then you'll have thermophiles or hyperthermophiles that will live in very, very hot environments that really run the gambit in temperature. What you're going to find is these different species of bacteria have evolved highly different enzymes that are more tolerant of these different temperatures. At the lower temperatures, they function better. They have sort of a lower optimal rate, optimal temperature. But like at those lower temperatures, they're just more functional than they would be for something more like this one here in this image. Other species in the hyperthermophiles will be way over here, far beyond the temperature that many enzymes will denature at, and they'll still be functional. And there's a lot of flexibility there that has a lot to do with the composition of those enzymes, like what they're made of, what shape they have, and also other features of the cell itself. They've evolved various strategies to help assist their cells functioning at these very, very, very high or very, very, very low temperatures. Ultimately, this comes down to the shape of these enzymes, what I'm really talking about here. So the shape here is the decider for an enzyme's function. Not necessarily entirely its ability to survive different temperatures, but its job. What it is specifically doing, what it is reacting with, and what it is doing with those molecules, what it is turning them into. So the lock really has to fit the key here, okay? The shape is so important for that. Enzymes acquire their shape a lot of different ways. There's a lot of things that go into this equation of why they have the shape they do. Of course, you have the sequence of their amino acids. You also have the interaction of those amino acids with the environment. That might mean that the way that they're interacting with water, some of these amino acids are nonpolar. Some of them are very polar. Some of them are ionic, where they're actually ionized, and so they want to interact with water much more. Other amino acids will be associating with water in interesting ways, such as creating those hydrogen bonds with them, which makes them those parts of the molecule anyway, it makes them hydrophilic. They love associating with water. Other parts that don't like water so much, they become hydrophobic. They won't associate with water at all, and those parts will be curled tightly inside of the center of the protein, trying not to interact with any other molecules but other nonpolar molecules. And then you'll also even have interactions between different amino acids. So amino acids will even reach across the protein itself and form brief bonds or much more long-term bonds with each other. So you may have more short-lived hydrogen bonds that will help hold parts of the molecule together, and then you'll even have sometimes disulfide bonds that form. So two parts of the molecule that have free sulfur that wants to reach over and bind, create a sulfur-sulfur bond, for example. And so for some of these interactions, high temperatures will disrupt them. It'll make hydrogen bonds much weaker and make them harder to form. As soon as they form, they break, and that's the end. And so they don't really have that sort of connection, the physical bond that holds them, or they can disrupt the hydrophilic hydrophobic interactions a little bit and different things like that. So these disruptions can change the shape and therefore the function of the protein without ever coming in and breaking this protein apart. It's important to remember that the protein will always remain very, intact as it reacts with these higher and higher temperatures. It takes very, very, very high temperatures in order to fully break the covalent bonds that hold individual amino acids together in a protein. So you're not really going to see that. Instead, you're going to see this more complicated shape unwind and just become a loose ribbon. Okay, where now it does not have the function that comes from its original shape. Just like enzymes have an optimal temperature, enzymes will also have an optimal pH range. And unlike temperature, which is pretty interesting, unlike temperature, proteins will denature at excessively high pHs and excessively low pHs, meaning if the environment is too basic, too alkaline, or if it is too acidic, it will change the shape of the enzyme and make it lose its function. Okay, so that's really important for like the function of these enzymes, because even slight deviations in the pH of their environment can actually reduce, just like with temperature, can reduce their efficiency and move them closer to denaturation. Okay, so it is really important to remember this as well. So the way this really works is the pH is interacting with the individual amino acids and their charge. It will react with parts of these amino acids and potentially ionize them, which makes them more reactive. It makes them want to bind or interact more with other things. And ultimately, this will force it to change the way that it interacts with itself, with its environment, with water around it, different things like that, which will lead to a change in its shape, right? Now, most enzymes function best at a neutral pH across, all of biology. Basically, you have the majority of organisms and their enzymes function best at neutral pH. But this is really just true for neutrophiles, right? Organisms that prefer a neutral pH. That's the majority of organisms that fall sort of within the happy medium that we find most places on Earth. So this changes, though, for extremophiles, organisms that love those extreme conditions. You can, just like you have psychrophiles and thermophiles, you can have acidophiles and alkalophiles. Some of these terms are just hard to say sometimes. Alkalophiles. So, so these different species that have evolved to live in these environments, just like the thermally adjusted or evolved species, you have these extremophiles that have evolved to live in these extreme pHs. They also have a variety of adaptations to help them survive in those conditions as well. Their enzymes are themselves a bit different. They function quite differently. So the other really important aspect of what influences enzyme efficiency, besides temperature, besides pH, is the concentration of the substrate itself. So there's going to be a finite number of enzymes and also a finite number of substrates to react with those enzymes. So at a low concentration, if there's a lot of enzymes and a low number of substrates, the reaction rate is it's going to be slow. Because the substrate is going to have to try to find the individual enzymes, right? For the set number of enzymes that you have, if you have too low a number of substrates, they are just bouncing around due to sort of the chaotic interactions between molecules at this small level. Molecules will, because they are energetic, they contain some energy, they're moving around, they will knock into each other and knock other molecules away in different directions. It's kind of like an infinite game of pool where the balls are the enzymes and they just keep on knocking into each other infinitely. So in this process, this all occurs inside of a cell, inside of this confined space, inside of this fluid environment. But now the enzymes have to actually find the substrates, or the substrates have to find the enzymes in order to complete this reaction. So at this point, the reaction rate is quite low. It becomes less common, less frequent for the substrate and the enzyme to collide, and certainly collide in the right way for the reaction to happen. But as you increase the amount of the substrate, as the concentration increases, so too does the rate of reaction. Because now you're adding more substrate, there's more substrate bouncing around in the medium, it becomes more likely that those substrates are going to interact with one of the enzymes. And this increases to a point, because eventually you reach a point where there are more substrates than there are enzymes, and at that rate, it basically, the enzyme is all reacting. Everything is reacting, so it can't react any faster. All of the enzymes are active, but there aren't any more enzymes in there to act more. And they also can't change the speed of their reaction. The speed of reaction just happens the exact same for any one enzyme. So once you cross that threshold, you're not really gaining any more speed of reaction, right, across this system. So just to recap, starting down here, keeping enzyme concentration, keeping enzymes the same, right? You'll have this relationship where as substrate abundance increases, so too will the rate of reaction. Okay, you'll add more substrates, and you'll increase the rate of reaction to a point until there's a tapering effect, and eventually you reach what we call an asymptote, where really you're not getting, we're at a stable point. We're really not reaching this sort of maximum threshold that's possible. You get a tapering off. Okay. The same thing, by the way, is possible if you switch things around and you say, instead, I'm going to keep the substrate concentration the exact same, and I'm going to increase the amount of enzymes. It's the same kind of equation here, right? The more enzymes in the solution, the faster the reaction, until a point where all of the substrate is being reacted, right? So let's talk again about enzymes and their usefulness. Okay, are enzymes useful? Yes, unquestionably. But are they necessary? Well, technically no. These reactions, all these different reactions, can technically occur without enzymes, but they happen at incredibly low frequencies. They happen very, very, very slowly. Very slowly. Too slow for life to function efficiently. Okay? Not to mention just the amount of energy that's needed to make those reactions happen in the absence of these enzymes. You need different conditions. So the enzymes technically are a necessary element. They make life happen at the speeds that we're familiar with. So enzymes and these metabolic processes are going to vary considerably across species, which makes them very useful as diagnostic tools to help us narrow down a species identification, for example. So some of the things that we're going to be doing in our unknown lab is going to be testing the enzymatic activity, the metabolic activity of different species, because that's going to vary so much across different species. And it can vary medicinally and ecologically in some very important ways. Metabolic variation can also account for differences in a species' vulnerability or resistance to an antibiotic. It might even associate with their ability to produce antibiotics, because it's important to remember that many species of bacteria will also in turn be making their own antibiotics, but antibiotics that don't hurt them as much as they hurt their neighbors, so that they can get an advantage over bacteria in their area. It can affect the ability for these organisms to digest certain resources or utilize them. We've already kind of seen that species can vary in their ability to digest lactose, to mannitose, different things like that, or digest things under certain conditions as well. And then various kinds of chemical production, okay? Various different things that bacteria may be producing, such as those toxins, such as those defense compounds, that just simply may be present in one species and not in another. We can even actually harness this capability for ourselves as we try to develop different kinds of medication and find more efficient ways to make different drugs, different things like that. So we can actually today, we can take the genes for specific enzymes that we're interested in, for specific enzymes that will make certain things that we're interested in, even if we don't want the enzyme itself, but we want the product that the enzyme makes. We can actually take the genes that will make those enzymes and insert them into a bacterial species like, for example, E. coli. And then from there, we can have that species make the compound for us. And we can just extract it from them whenever we want. This is such an amazing piece of technology, I can't even explain it. I can't even do it justice. It has revolutionized the lives of many people today. So we, for example, if you have had insulin injections at any point, that insulin was made by E. coli or some other species. There's a ton of different compounds like this that we have simply taken the genes of that are responsible for making those things, and we've stuck them into something that's really easy to grow in the laboratory. And it's changed the game in a lot of ways. So let me give a brief example about methods of diagnosis and how enzymes can be diagnostic. Okay, there are some species that are going to look really, really similar to one another, maybe even identical, but their metabolism differs dramatically. So they have different kinds of metabolic pathways. So, for example, Streptococcus pyogenes and Staphylococcus aureus are a good example of this. These are both coccoid bacteria. They're both gram-positive. Okay, they both, they differ in their cell arrangement, where Staphylococcus experiences the staph-like clumps. That's very associated with Staphylococcus, and then Streptococcus forms chains like this. But when you have so much of the stuff, it becomes kind of hard for you to easily differentiate, this is a strep type arrangement, and this is a staph type arrangement. You might not be so confident in that, okay? But what they do differ meaningfully in is their ability to break down hydrogen peroxide. As we explored before in a previous chapter, hydrogen peroxide is extremely toxic. It is really detrimental to life generally, to bacteria of all kinds, certainly. But some of these bacteria have enzymes they can deploy to quickly break down the hydrogen peroxide so that they're not hurt by it. But some species have this, some do not. Okay? So when we administer hydrogen peroxide to a culture and we see if it's going to break it down. If we don't get a reaction, okay, they lack the enzyme that is associated with breaking this stuff down. It's called catalase. If you do see a reaction, right, as we do here, the bubbling, furious bubbling, this means that the bacteria does in fact have the enzyme, and the enzyme is actively breaking down hydrogen peroxide. Now, hydrogen peroxide still works as a method of sterilization, right? A method of antisepsis of a wound, because bacteria can only defend against so much of that hydrogen peroxide. If you really flood an injury with hydrogen peroxide, you're going to see a lot of bubbling. Some of that may be because of bacteria. A lot of it is because of your tissue, because you also make catalase. And so because hydrogen peroxide is bad for us, we also need to break it down if we're ever exposed to it. So for that same reason, we will make that stuff. So our wounds will also bubble quite a lot with regard to their exposure to hydrogen peroxide. But the bubbling does not necessarily mean that the bacteria has successfully made itself safe. But it is part of its defense. Usually bacteria aren't encountering such large quantities of hydrogen peroxide. It's not exactly normal to encounter it in such abundance. We'll say that. But it's important to note that these enzymes don't work all the time, right? And we also don't want them to work all the time. Enzymes are a kind of thing that we want to work on demand. We don't need the products of every metabolic pathway all the time. Instead, you only need it when you need it, right? So, sometimes other metabolic pathways are going to take more priority. Sometimes you're going to need to do one type of reaction to save your butt, and other reactions can take the backseat for a little bit. For example, if you're being bombarded by hydrogen peroxide, well, making more catalase could be the difference between life and death, and making that catalase active, and so that it is out there working very actively. And so, other things like eating, like digesting, like making more of yourself, making your defense compounds against antibiotics, or maybe making an antibiotic yourself, well, those things fall to the wayside if your need of another thing takes priority. And so, to accomplish these tasks, organisms, again, this is something that's universal, not just bacteria, but all life, are going to modify their enzymes in a few different ways to make them active sometimes and inactive other times. And there's a lot of ways you can do that. First, you can just simply produce more or less of that enzyme. So if you have a bunch of it and you don't need as much now, you could just simply stop making it for a little bit and reduce the number of enzymes you've actually produced. If you don't want to do that, or if you have another strategy, you could also be changing the availability of the substrate itself. So again, remember the rate of reaction is dependent on the concentration of the substrate, right? So we can change that by reducing the availability of that substrate in the cell. Then we can also directly modify the enzyme itself. We could take the enzyme, and if it's an allosteric enzyme, we could add an allosteric modulator to inhibit it, or if it's maybe it's an apoenzyme that has been active for a while, we could unbind a cofactor from it. We could strip a cofactor from it and make it non-functional again. So there's a few different things you can do. There are more things too, right? So there are some metabolic pathways that organisms have where they have what's called a feedback inhibition. Okay, so what this really means is that you'll have this metabolic pathway, you'll have a series of reactions, So, reaction 1, reaction 2, reaction 3, and you get this end product. The end product, in turn, comes over here and it binds to enzyme 1, and it changes the shape of enzyme 1 and makes it unable to bind to the starting substrate to make that end product. So essentially what's happening is the products of that reaction stop the reaction once it's made. Okay? It deactivates the system once it's been done. So this is really useful. This process is super useful because it basically prevents you from making like an infinite number of a compound, but you only really need a little bit of it. And you can still keep all the enzymes out and about. You don't have to change the amount of the enzymes that you've made. You don't have to go and destroy any enzymes. You just turn them off. You turn them off with the same stuff you used to make them, that you used them to make. A really good example of feedback inhibition is with E. coli and isoleucine. So isoleucine is an amino acid. Okay, we can get it from our diet. We can also some organisms can also make it. Okay, so excuse me. So in E. coli, there is a metabolic pathway to make isoleucine, and isoleucine is really useful because it's used to make a lot of proteins. But there are many different kinds of amino acids used to make proteins. You can't just rely only on isoleucine. And you certainly don't want to take, a bunch of other things, maybe other amino acids, other types of amino acids, and react them through this pathway to make a bunch of isoleucine. And then you don't need the isoleucine at the end. You only need a little bit of it. Okay? So E. coli, for this reason, has a feedback inhibition pathway in its isoleucine synthesis metabolism. So when the cell needs to make isoleucine, it begins the process of reacting all these different things. The enzymes get to work making isoleucine. Then, when isoleucine is made, when it has reached a certain abundance where loose isoleucine is bouncing around, it can collide with the starting enzymes in that process and deactivate it, right? It serves as an allosteric modulator, right? It can deactivate, it can inhibit. It can inhibit the species or the enzyme, right? So essentially what's going on here is when isoleucine is abundant, there's enough of the isoleucine that's loose to bind as an allosteric inhibitor to the enzyme to make isoleucine. When it is scarce, those inhibitors are gone, and the enzymes are allowed to start making isoleucine again until it becomes common enough, abundant enough, that it again binds with that original enzyme and deactivates it. This is what this looks like, by the way. Of course, I don't really need you to remember any of this except for sort of what is happening here. So threonine is another amino acid. Okay, so we take this and react it, and we get isoleucine. But we may very well need this process, right? We may need this process, and so isoleucine can in turn go back over here and bind to and deactivate the enzyme responsible for starting this reaction, halting it, preventing it from moving forward. This whole thing over here, this pyruvate to valine process, this is another metabolic pathway, and the product of the valine can actually come over and also deactivate this as well. So there's also many different methods. You can have feedback inhibition, but you can also have inhibition from other products as well. So enzymes can ultimately be inhibited by lots of different stuff. It's not just cofactors. It's not just allosteric inhibitors, that kind of stuff. It's not just feedback inhibition products either, right? So there's lots of different ways this can happen. You can have what we call competitive inhibitors. So a competitive inhibitor is going to come in and bind on the active site where the substrate would normally bind, right? And the competitive inhibitor is a molecule that isn't going to be broken down by the enzyme. It's basically just plugging the hole. It occupies the space. It prevents the substrate from coming in and binding, and so there is no reaction. This just occupies space, right? Totally blocks the actual site, the active site, okay? But we could also, so we call it, by the way, competitive because the inhibitor is competing for access to the binding site with the substrate. So these two both want to bind here, but the competitive inhibitor comes in, binds, and it is more competitive. It kind of beats out the substrate, and it occupies that space for some amount of time. It varies a lot, depending on the molecule, depending on the enzyme, different things like that. On the other hand, you can have a non-competitive inhibitor, and this is when you have an inhibitor that's going to bind to a location that is not the binding site. So some other allosteric binding site. It's going to come in and bind. It is not competing for that active site, so the substrate can still bind, but there's no reaction. Or maybe the enzyme can't even bind at that point, where the attachment of that non-competitive inhibitor changes the shape of the enzyme and prevents it not just from reacting, but from binding at all. So there's lots of different ways that this might happen. A cool example of this is sulfanilamide. So sulfanilamide it's an example of a competitive inhibitor and an antibiotic. So this is the type of antibiotic that will come in and compete with bacterial enzymes in their efforts to build folic acid. Okay, so folic acid is a necessary B vitamin. It's something that all life needs, and it's something that is really important in the process of making more DNA and RNA. Okay, so if you don't have the folic acid, well, you're not replicating your DNA, which means you aren't doing mitosis or any kind of cell division, right? You're not dividing yourself at all if you don't have the materials to do that. So this is what makes sulfanilamide a really effective antibiotic. It is very much stopping a critical thing that the cell wants to do to continue to be a problem for us. So in this case, this is the reaction, okay? So you start out with this PABA compound, okay? This para-aminobenzoic acid is what it is. And it is going to react using this enzyme as a catalyst to create, essentially go down this pathway to make folic acid, okay? But without this, with this sulfanilamide attached to it, okay, it comes in, it attaches the exact same site that the PABA attaches to, okay, and so it prevents the binding of PABA, right? Look at the similarity in the shape of these two molecules. They look incredibly similar, right? Incredibly similar. This is what I mean by lock and key and how similar shaped keys can fit in the same lock. They just may not necessarily have a reaction once they fit there. So here, sulfanilamide, incredibly similar in shape to para-aminobenzoic acid. It comes in, fills the active site, and it just sits there. It doesn't go away. It just stays attached to this enzyme. And the progenitor for folic acid never gets the chance to react. It never gets the chance to be turned into folic acid, which means that the cell can't replicate its DNA, which means that the cell cannot replicate, cannot actually reproduce. in effect, stopping its rapid growth, right? That's what makes this sulfanilamide a very effective antibiotic. And the cool thing about this is that human cells were actually not affected by it because humans don't make folic acid. We get it entirely from our diet. So there's no real worry. We don't actually have an enzyme to make it. We're not dependent on that in order to get our folic acid. And so it can hang out in our body and it doesn't really do anything. We call this being selectively toxic, okay? Selectively toxic, where it only is toxic against some organisms. In this case, toxic only against the bacteria that we're using to target it with, okay? And not against us. So, okay, now it's time to explore a pretty complicated and very important aspect of our metabolism, and that is any metabolism really, and that is glucose and ATP metabolism. Okay, so this process, the process of taking glucose, breaking it down, and turning the energy inside of it, using that to build ATP molecules, is super important. Right, it is the basis of a lot of life. Right, a critical process that everything does. Think back about catabolism and anabolism here, okay? Think back about this. So we should also see that we can kind of figure this out where catabolism, we're talking about the breakdown of those glucose molecules, and anabolism, we're talking about the assembly of ATP molecules, right? So there are a few ways to do this process, and they involve oxygen and other methods that don't use oxygen. In this way, we have aerobic and anaerobic respiration processes. Okay, these pathways that will use or not use oxygen. So let's explore some of them. And before we do that, this is what ATP looks like, right? I think that I've drawn this in class before. I can't quite remember, but this really is where the energy is located here. Why this molecule is so useful. You have this chain of phosphates that are attached at these intermediate bonds, but this last one is a pretty high energy bond that, when broken, is going to release a lot of energy. And that energy is going to be released as a combination of physical kinetic energy, where this phosphate and the rest of the molecule are going to be pushed away from each other pretty quickly. And then also as thermal energy, which is just a measure of the speed of molecule movement. Okay, and generally this process of making ATP from other things is pretty straightforward. It's a cyclical process where you have ATP. ATP can break and release energy that's stored inside of it, and it forms ADP, right? Adenosine diphosphate instead of triphosphate, and a loose phosphate group, that one that gets thrown away. But then we can actually build ATP again by taking those two parts, using energy to push the phosphate back in place and form a bond here, and create ATP again. Okay? So this process is, it does require energy. I want to be clear. We use energy in the form of glucose and also other sources too, at times, to make ATP. We will even use ATP to make ATP. Okay, we can do that too. That is very assisting for us. It's basically like you got to spend some money to make some money, right? You got to spend some ATP to make some ATP. This is not a perfect system. Energy is lost with every reaction. So it is lost to the environment of the cell, which is then lost to the environment outside of the cell as it trickles out. Okay? So during the breakdown stage, we're releasing energy, and some of that is escaping, but we're storing some of that energy as ATP when we then go to make ATP from those resources. Then when we use ATP, okay, we use ATP, release energy, We capture some of that energy to fuel anabolism, but some of it is also lost in the process. Okay? This is essentially that process, what that looks like. And I really want to stress, it is not a closed system entirely, because you will be losing energy like constantly, which is why it is so important to be getting resources from the environment, to be getting new energy sources that can be turned into ATP, because it's not a perfect cycle. All right, now let's talk about this process, all right? You're probably familiar to some degree of this process in humans. Starts with glycolysis, okay? This is an anaerobic process where we take glucose and we convert it through a series of reactions into pyruvate. And in the process, we make a little bit of ATP. We had to spend some ATP in the process to do it, but we got more than we spent, okay? It's not very efficient. After that, we go to the Krebs cycle, okay, which is also an anaerobic process, but typically we don't really do this unless we're ready to do the next step, which is an aerobic process. So that's the Krebs cycle. The Krebs cycle is also called the citric acid cycle, which you may have also heard of. This is a cyclical reaction where you take pyruvate, turn it into acetyl-CoA, and then react that a bunch of times until you produce lots of interesting stuff. A little bit of ATP, but also some important other molecules that are important for the electron transport chain, which is the last step in the process. Okay, this, for humans anyway, is the aerobic part of the process. This and the Krebs cycle for us occurs in the mitochondrion. Glycolysis is something that occurs in the cytosol. Electron transport chain, I'm going to show in a little bit, but essentially what it does is it will shunt a bunch of hydrogen ions across the membrane and hold it there and use it as a power to essentially turn a crank and push ADP to P and force them to react. This is a very efficient process, and we can create tons of ATP this way, which is very useful. Okay, so these processes that I just explored here, they fall into two main categories of ATP synthesis. Okay, glycolysis and the Krebs cycle are doing what we call substrate phosphorylation, substrate phosphorylation, which is where we're taking P's, loose phosphates, and attaching them to ADP using some other substrate as a donor, right? Not ATP necessarily, but other things. We're stripping the phosphates off of those things, okay? Then we have oxidative phosphorylation. Oxidative phosphorylation is when we are attaching phosphates to ADP, using, I mean, certainly the P's are coming from somewhere, but the power that facilitates this is coming from like free energy, right? Free energy that is generated in this environment from what we call a redox reaction, okay? Now we're dipping into chemistry again, heavy chemistry influence here. Redox reactions, which you may remember from chemistry, this is oxidation reduction or reduction oxidation reactions, right? Re for reduction, oxidation for, or ox for oxidation, right? These types of reactions are about loss and gain of something, okay? The loss is oxidation. The gain is reduction. I realize that sounds kind of counterintuitive, but it does make sense. It does make sense in chemistry, okay? What we're talking about here when we talk about loss and gain, we're talking about atoms or most commonly electrons, okay? Usually electrons, but I think really in the case here in oxidative phosphorylation, we're seeing loss and gain of individual atoms, right? That is also happening here. So there's a useful useful naming. No, what's that called? I don't know what it's called. When you got like a a way to remember these things. I always forget what those are called. The little games that you can sort of set these things up where one word means a whole bunch of stuff. Whatever. So this one is called oil rig, right? Oil rig. Oxidation is electron loss, right? Reduction is electron gain. Okay, oil rig. You can also think of it like oxidation is loss, reduction is gain for the purposes here. Okay, since we do see reduction and oxidation of atoms as well. Okay, let's look at some of these processes a little bit, right? So usually these methods of substrate phosphorylation begin with carbohydrates. Okay, you can use other things, but usually these things start with glucose. You can use other stuff. You can use other carbs. You can even use like lipids and different things like that. But you have to react with them a bunch of times to turn them into something that can be used in this process. So a lot of the time, even though you have lots of different things that could be used to do glycolysis, you have to reach the starting point of glycolysis, which is using, getting glucose, right? Getting glucose in some shape, okay? So here's a great example of that, which is galactose, okay? Galactose is a different kind of sugar. It is, on the surface, very similar in appearance to glucose, right? The real difference is just the location of the H and OH bound to this carbon. Okay, seems insignificant to us, but it really does influence the ability for this for galactose to be used in this reaction. So, in order to take galactose and send it into glycolysis, it has to go through these additional steps here. Okay, you start with galactose, you react it with galactokinase, and then you get galactose 1 phosphate, right? So this is, remember, kinase is the attaching of one phosphate group to another molecule. Okay, so we can figure out exactly what this name is by looking at it. Galactokinase. Galacto like galactose, kinase like attaching a phosphate group to a thing. Okay, you take the result here, you react it again with a different kind of enzyme, you get glucose 1 phosphate. Now all of a sudden we've reached glucose. We can take that, go through the process. There's an enzyme that's part of glycolysis that will react it and create glucose 6 phosphate. And now you're ready to do full glycolysis. Okay, full glycolysis now. So again, the gist of glycolysis, this is an anaerobic respiration pathway. Anaerobic respiration pathway. The result of the reaction here is you take these simple sugars and you turn them into 2-pyruvate, right? So you make a cut here and a cut here, and you end up here after a series of reactions, multiple different reactions with 2 pyruvate molecules, 2 different pyruvate molecules. In this process, you're also taking a specific molecule called NAD+. Okay, NAD+. This is an important reactant in this glycolysis pathway, and you're using it up. What I mean by using it up, you are adding a hydrogen onto it. Okay, you're adding a hydrogen onto it, so you're reducing it. So this is really important, though. We'll see this molecule come up again at a later time here. Okay, keep an eye on it. The process will produce not just 2-pyruvate, but also 2 ATP on top of what was, well, you have to spend 2 ATP to produce 4 ATP. So you end up with a net 2 ATP, if that makes sense. This is the series of reactions for glycolysis. You don't need to remember these. This is not that class. But I wanted to show you the series of steps in order to reach this point. keep in mind there is an enzyme mediating each one of these processes. These letters here, right? HK, PGI, PFK, ALDO, GADPH, GAPDH, sorry, all these different things. These are the enzymes. They're abbreviations for the enzymes that that their job is to regulate this specific reaction. And here are all the names written out, actually. So, there's a lot of steps involved here to get here. So there's many different enzymes that are all working in sync with one another in order to get to the pyruvate, at the end. So, it is important to remember here, okay? Then we have the Krebs cycle, okay? Again, this is a process that technically does not involve oxygen, not really, but we, cells don't usually use this thing, ours or bacteria, unless they are about to do oxidative phosphorylation. Okay, so the Krebs cycle here, what it's going to do is you're going to convert the pyruvates that came out of the last reaction into this compound acetyl-CoA. Okay, you get the pyruvates or pyruvic acid up here. You react it to create acetyl-CoA. Look what's happening again during that process. You have more NAD plus that is reduced, it becomes NADH. Okay, and then you get this cyclical reaction that happens like this. Okay, and it goes around and around a couple times. During the process, you can make ATP. You really just only make one ATP per cycle. Okay, so it is not especially efficient at making this stuff. You are also generating more of these interesting hydrogen donors and receivers. Okay, here's another NAD plus getting a reaction, is getting reduced. Here's an FAD that's getting reduced into FADH2. Okay, so here again we've got more of these hydrogen receivers acquiring hydrogens. More of them over here and here. Okay, keep this in mind for as we approach the next steps. The results of this process, okay, the results of this process are the generation of 1 ATP per round, 3 NADHs per round. That shouldn't actually have a charge to it. That's a typo. And then 1 FADH2 per round. Okay, really not getting a lot of ATP produced during this time, but that's okay, because the next step is much more productive for this, if the cell is going to do that. Okay. So this right here, again, drawing your attention to these special molecules, the NADHs, FADH2s. Okay. The process to get them is to hydrogenate them, right, or reduce them. So we are adding hydrogens onto them. Over here, though, right, we can also take them off. We can remove them and isolate just the hydrogen ion, which is effectively just a proton. It's highly charged. Okay, so oxidation can remove them. And this is the electron transport chain. That is what happens here during this process. And a very critical aspect of the electron transport chain's function is the stripping of hydrogens off of those NADHs, off of those FADH2s, and in the process can kind of set these molecules back to their original position. Okay, their original charges, and the original state of lacking those hydrogens on them. Okay, so for us, this is something that occurs in the mitochondria, right? So our organisms of eukaryotes will do this in their mitochondria. In an organism that is prokaryote, of course, they lack a mitochondrion. Well, this process occurs on the cell membrane, okay, on the just like inside the cell membrane and across it. All right, it's really interesting this process. So and the location that it occurs. Remember that the cell membrane for a bacterium is just as much about containing the contents of a cell as it is about generating energy, because the whole thing is covered in that electron transport chain. Okay, it's all over the place there. So what is happening here? Well, inside of the membrane, you have a bunch of these embedded proteins. And these proteins will come over here, they'll strip the hydrogens off of these NADHs and the FADH2s, and they push them across the membrane from the inside of the cell to the outside of the cell. In the space, in eukaryotes, it's the space between the inner membrane and the outer membrane of the mitochondrion. Remember, the mitochondrion has two membranes. For a cell, it is the space, For a bacterial cell, it's the space between the cell membrane and the cell wall. Okay? And in that space, all of these hydrogen ions start to collect. Okay, you get this building charge here because all these molecules are doing this. They're all, pushing hydrogens across these membranes using these specialized proteins, and along the way, they're also stripping electrons. Okay, and the electrons will also be passed right through these different proteins all the way to this last one, where in the case of organisms that use oxygen to do this, you get this reaction where I'm skipping a step here. Essentially, this enzyme right here is ATP synthase, right? The synthesizer of ATP. It's the only path for the hydrogen ions to flow back into the cell, okay? To flow back inside the cell. Keep in mind, the cell is building high concentrations of the stuff here and keeping very low concentrations of it here, right? Diffusion hates that. These processes of diffusion really, really, really dislike having these strong charge differentials or high concentrations of one thing here and very low concentrations of that thing here. So the hydrogen ions want to come back across the membrane. They want to distribute themselves evenly on either side of the membrane. So half here, okay? But they can't achieve that because they can't pass freely through this membrane, right? Charged particles do not pass very well through the lipid membrane, okay? Phospholipids won't let them through. So the cell creates spaces where they can pass, little pathways here, and that one of those pathways is through ATP synthase, right? So essentially, hydrogen ion comes in, it passes through here, kind of like passing down a river that has a water wheel on it. And as they pass through, they push the water wheel, just like water would on a normal water wheel, okay? Inside of that water wheel, you have ADP and a phosphate that is sitting there waiting to be pushed together. So the power of diffusion is pushing the hydrogen ions through this space. The hydrogen ions are pushing their way and turning the water wheel. That is what pushes ADP and P together to form ATP. Okay? There you're getting the building, the construction, the synthesis of ATP, right? Hey, we've done it. Then, that hydrogen ion, well, it has to go somewhere. Those hydrogen ions, they're very reactive at this point. They're going to pick up these two loose electrons that came from over here. Okay, they're going to come pick this up, so they'll stop being ionized and they'll react with oxygen to form water, right? So this is where we are getting the oxygen coming in. It's an important part of that process that is sort of helping to stabilize these ions. Okay, it's finding a home for these hydrogen ions. So again, remember this works because of diffusion, right? That the cell is fighting against diffusion, against equilibrium. And because the ions really want to be evenly distributed. They want to reach equilibrium, just like this GIF over here, right? This is what they wish they could be doing. And this right here is what they wish they could be doing. They wish they could be moving from this area of high concentration to this area of low concentration, okay? But they can't. They can't pass through this. Only water can pass freely through these membranes. Charged particles can't do this very easily, okay? So they bounce on the surface and they're unable to pass through. That's why the cell provides a singular path in order to allow them to pass through. And as they do so, they use them to power that process of oxidative phosphorylation. So ATP synthase is that last step, and I wanted to show an animation of this because it's really useful to see the thing in action. Okay, it's hard to sort of imagine it otherwise. So this is what it looks like. You've got the ion very clustered on this side, and there's only one path through. Okay, and as they push through this protein here, they cause it to spin in its anchor. Okay, it's stuck in this membrane. They cause it to spin in the membrane, and it turns this wheel here that pushes ADP and P together. Okay, and it spits out the rest of the those hydrogens over here to react with oxygen and then become stabilized. Right. So this is what that process looks like. We can see here in this animation that we've got all these little dots that are bouncing around. Those represent the hydrogen ions that are super concentrated over here and they really want to pass through. They're doing their best to pass through, and they're not really able to until they encounter just the single space, just the right space in order to pass through it. And we can see them here. That was one. That was one. So as they pass through, they turn this wheel. Okay, they cause this wheel to turn, which in turn pushes these compounds together once they're inside, right? So the big one, the blue one here is the ADP molecule, and then that little green one is the loose P, right? They come in, they fit in place, and then the wheel turns, and it causes them to be pushed together, okay? So this is another animation that I really like that shows the whole process at work here. Okay. Again, this is the inside of the cell. This over here at the bottom is the outside of the cell. Top inside, bottom outside. The arrows are showing the movement of hydrogen ions. They're passing through these proteins, passing through them, and then only allowed to pass back through in this wheel. Okay. Only in the wheel. That causes this thing to turn, and this a big enzyme complex here to change shape, making them react. And this is something that occurs across the entirety of the cell membrane. Okay, it's important to remember it's not just like, oh, it's just a few enzymes here and there. They're all over the place. Think back about how I described that there's lots of different things that may be anchored in a cell membrane. Many different things. Many different proteins. Some of those proteins may serve some functions like helping things move across the membrane. Some of them are involved in a process like this, like ATP synthesis, okay, electron transport chain. As I said before, this process is going to be really productive. This is going to produce upwards of 38 ATP per turn, okay, or per run. So that, and by that I mean like the products of one glucose molecule can down the road produce this much ATP, which is really impressive, right? But importantly, it's very different, very much more effective than glycolysis or the Krebs cycle on its own, which only produced 2 net ATP and 1 ATP, respectively. Okay? But it's important to note that this is a process that requires oxygen for most organisms, right? Most organisms that do this are going to be doing this with oxygen. That's sort of like the dominant type of life today is like a life that uses oxygen. So oxygen becomes that important electron acceptor together with the hydrogen. So without it, there is no oxidative phosphorylation. It needs it in order to do that process. But there are some organisms that do other things, right? Aerobic organisms will do all three processes. Okay, we'll do glycolysis, Krebs cycle, electron transport chain. Some organisms that are anaerobic will only do glycolysis, and they'll just do a ton of glycolysis. Okay, or some organisms that are facultatively aerobic, facultatively anaerobic, that they can survive anaerobically, but would prefer to do oxygen. Under those circumstances, they may be just doing glycolysis over and over and over again. Okay, and so these organisms can actually switch up. They can switch up those tactics when oxygen becomes scarce. We see that, for example, in yeast as we brew beer, for example. Bacteria, though, get pretty interesting in this regard. So there are some anaerobic species that they do anaerobic respiration, but they also utilize the electron transport chain. In doing so, they just don't do oxidative phosphorylation. They do other kinds of phosphorylation that use something else as the electron acceptor, not oxygen. Something like nitrate or sulfate or carbonate, some other molecule that will otherwise receive those materials to sort of like solve that problem. And this is a pretty cool adaptation. It's a nice adaptation to utilize different resources because oxygen, again, is very toxic. It's really dangerous to a lot of life, especially anaerobic life. And being able to use the electron transport chain instead of just glycolysis forever is a game changer. It can, we just explored it. It can produce staggering amounts of ATP, which is really impressive, right? This method, right, the electron transport chain is not exclusive to aerobic organisms technically, okay? And it's important to note also that this anaerobic respiration pathway can, in these organisms, be just as effective as the aerobic respiration pathways, which is really cool, producing very similar quantities of ATP. For anaerobic organisms that are not doing that, though, that are not doing the electron transport chain, they have to face something difficult, which is that the materials that they used to do glycolysis, well, they used up a lot of that in the process of running glycolysis, namely those NADHs, the NAD pluses, I mean. So you're taking NAD pluses, you're reacting them in glycolysis to make two NADHs. Normally, those NADHs would go on to be an important role in the electron transport chain. They give up their hydrogens, and they become hydrogen ions that can, and they also are like, they're electron donors as well. So they are very important in those stages, but they don't now do anything in glycolysis in this state as NADHs. Only NAD plus, right, will actually react in glycolysis. So how do you get these resources back? You don't really want to make more of them from scratch. That sucks. That's very wasteful. So instead, these organisms have devised a strategy called fermentation. Okay, and so this is a process that will oxidize NADH. It'll take that hydrogen ion and strip it off, and so it'll be NAD plus again. And that way they can repeat glycolysis without ever needing to make more NADHs, or make more NAD pluses, I mean. It's a process that doesn't use ATP, okay, which is really useful because you're not using the same ATP that you just made. and it produces some waste material in the process. Okay, it'll end up making some waste material from that pyruvate that was the result of glycolysis, if you think back here. So the key function here is really just replenishing NAD plus and sort of resetting the setup for glycolysis again. And so the way that this works is it's very similar, but also very different for a lot of different organisms, okay? Microbes are pretty cool with this regard because they will be making lots of different kinds of stuff as a waste product. They can take the pyruvate molecule and they can turn it into lots of different things, some of which is very useful to us. So, for example, there are some species that will be producing, like carbon dioxide. They'll produce unusual things like propionic acid. Lactic acid will be produced by some different bacteria. A lot of bacteria will do this, and some fungi as well, I believe. Saccharomyces and other yeasts are very popular for their production of carbon dioxide and ethanol as waste products. Ethanol is alcohol, so we use yeasts and similar species to brew beer and wine, but also leavened bread, different things like that. And then other species will make interesting things like acetone and isopropanol, which are quite unusual. So Clostridium is an example of that. Importantly, it's fermentation. We think about it as something that only microbes do. It's only something that species that we're growing for beer will do, or we're cultivating cheese or soy sauce or whatever, and we are going to use the products that they make. Okay, well, microbes aren't the only thing that do fermentation. We also do fermentation. Most life can do fermentation in some form. Okay, because there will be some situations, for example, where aerobic life may be having trouble accessing oxygen, and so the cells will do other activities. They'll still need access to ATP, but they kind of are limited in their ability to generate it because all they can do is glycolysis. So they still need to reset the system over and over and over again, so they don't just run out of resources to make it before they run out of glucose. So for humans, we produce lactic acid as our fermentation waste product. Okay, and we actually were pretty familiar with this because anytime you've gone to work out, for example, and you feel like that burn in your arms or your legs or your glutes or whatever, you feel that the pain, the ache, and even a burning sensation that may linger for a couple days even. And that is actually the lactic acid that you've produced. You've made this thing in your muscle cells. As your muscles were working, they ran out of oxygen, and your body couldn't bring those cells oxygen fast enough. So as a result, they had to rely on less effective methods to generate ATP, but they were still able to do their job, which is the important thing. Okay, so don't forget, it's a thing that we do too. And facultative animals or anaerobes are pretty cool because they have that ability to switch strategies. They can adopt aerobic conditions when appropriate, or when there isn't oxygen available, they'll switch to doing anaerobic methods as well. So there are some species that may be just as functional as an aerobe as they are as an anaerobe. Okay, so that's it today. Please make sure that you read the chapter as always, and come prepared for our next lab. Our next lab after this, our next chapter is chapter 9, which is all genetics. It's going to be a challenging subject, just like this is. So please study accordingly. Spend extra time preparing. And again, the next lecture is probably going to be long. This one was on the long side. I think the next one might be even longer. I'm not sure. But we'll see. All right, everyone. Have a good day.